Thank you so much for that kind introduction. I'd first like also to say thank you to the organizers for inviting me and giving me this challenging topic to speak on. It's been a really stimulating topic to prepare. So is alpha-synuclein still the basis for Parkinson's disease? And as I was preparing this, I thought most of you would just say immediately, yes, of course it is. And I was preparing it, and sometimes I said no during my preparations, maybe a surprise to some of you. However, I noticed by the time I finished my preparations, and you will hear exactly why soon, I began to think that maybe you would say stop and I would say go, go, go. And those of you who know your music history know that those are the classic lines from Hello, Goodbye with the Beatles. And the Beatles will follow us during this talk as we take a magical mystery tour of the role of alpha-synuclein, or not, in Parkinson's disease. So I'm going to divide this up first into three short sections that introduce you to alpha-synuclein. What is this protein? What's its genetic links to Parkinson's? And how does it have a role in the formation of Lewy bodies? So what does it actually do there? Then we will have 4 sections where I ask specific questions to see if alpha-synuclein is really, or I should say Lewy pathology is really necessary, and if it's sufficient for Parkinson's disease. Then I'll show you something about animal models, which I think are instructive and teach us something about the role of alpha-synuclein. And then I ask the question whether therapeutic strategies that are being tested tell us something about the role of synuclein in Parkinson's disease. And right at the end, we have the very important question, can we identify alpha-synuclein patients? So let me begin with a brief introduction to alpha-synuclein in the context of Parkinson's disease. Well, first of all, alpha-synuclein is expressed, obviously, quite a lot, both at the RNA and protein level in the brain. And I think everybody is well aware of this and appreciates this. And I think you also have heard that its precise functional role is not 100% certain, but it most certainly is in some way involved in synaptic vesicle recycling. So an important protein. Amazingly, removing alpha-synuclein in knockout mice has very small consequences on the behavior and the function of those mice. But this is what alpha-synuclein is believed to do in the brain. Now, if you look carefully at this diagram at the bottom, you can see that alpha-synuclein is also heavily expressed, present at the RNA and protein level in the bone marrow and in lymphoid tissues. And I think this is important. It's not going to be something I come back to, but I just want you to have this in the back of your minds because it's been suggested that alpha-synuclein has a role as an antiviral peptide. So it might be part of the immune system in the body's defense against pathogens, specifically viruses, possibly also bacteria. So maybe not something that will be recurring, but it's worth bearing in mind as we consider how alpha-synuclein, if it is involved in Parkinson's disease, how it starts the disease. So what exactly is alpha-synuclein? It's a protein made up of 140 amino acids. It has an N-terminal region where you'll see in this diagram that there are a few known point mutations that I'm going to come back to. Then it has the NAC region in the middle, and it has a C-terminal calcium binding domain. And what's special about alpha-synuclein, and we can look at it in a different way here, is that in its monomeric form, it is really rather non-structured. So it can adopt many different conformations that it transiently moves between, so to speak. You can see them here in this diagram. So that's not really why we think it's important, if it is important in Parkinson's disease. What makes it so special? It can be bound to membranes. I think I mentioned this. The a helical part of synuclein can be bound to membranes, but it can then be in an equilibrium between a more membrane-free form and the membrane-bound form. And the membrane-free form has the ability to form oligomers. Some of these oligomers actually then go on to form fibrils. So they're the so-called on-pathway oligomers. And they go through a small fibril stage called protofibrils. There are also other oligomers that are more amorphous, and it's not clear what they do. But these protofibrils then can assemble into large beta-plated sheets, which are the fibrils that we are familiar with in the pathology of Parkinson's disease. And it's interesting that there are different variants of these fibrils, and they can go by names such as strains, because they can affect monomeric alpha-synuclein and template, act as template, so that monomeric synuclein will then form oligomers and these specific shaped fibrils. So we're going to come back to these because they are important in the pathology. But I did mention something about mutations. So what is the genetic evidence that the 140 amino acids that make up alpha-synuclein are involved in Parkinson's disease? Well, there are, first of all, mutations, both in terms of missense mutations and gene dosage mutations that can directly cause neurological disease that is either virtually identical to Parkinson's or is a syndrome that has Parkinsonism as part of it. So this is important because these are experiments of nature, very rare, few families across the world, as you all know, but they tell us that in a dominant form of disease, these changes in alpha-synuclein actually cause neurodegeneration. That's not the only thing, because there is also the fact that amongst the now about 90 genetic risk loci that have been statistically associated with elevated or reduced Parkinson risk, amongst those loci, there is a locus that is very close to the synuclein gene, and it's one that's been repeatedly shown to be highly associated with changes in Parkinson risk. Now, such a polymorphism can regulate more than one gene potentially, but it is so close to the alpha-synuclein gene that it's very likely that it is involved in regulating synuclein levels. So maybe by regulating synuclein levels, it will elevate the risk for Parkinson's disease by there being more synuclein. And if synuclein crowds, it's more likely to aggregate. In a similar way to what I just showed you before, that if one has multiplications of the gene, if one has a duplication or a triplication of the gene, there is also this neurodegenerative disease with protein aggregation. So there is that strong genetic evidence linking alpha-synuclein, at least to rare familial forms of Parkinson's disease, and also regulating genetic risk in the garden variety of sporadic disease through those polymorphisms. So let's get to these aggregates that alpha-synuclein forms in Parkinson's disease. And you know they are really the main constituent of Lewy pathology. Lewy pathology, historically, we have to honor Fritz Jakob Heinrich Lewy, who was a student of Alois Alzheimer, who in 1912, for the first time, described the microscopic inclusions in neurons. And you see a drawing there from a publication he had in 1923. He described these, and they came to bear his name. Now, these were done with standard histological staining. Nobody knew really what they contained, so these hyaline inclusions. It wasn't until the landmark finding in 1997 in a team headed by Maria Grazia Spilentini and Michelle Gerdert that it became clear that Lewy bodies were primarily or are primarily made up of alpha-synuclein, and there's lots of lipids and other proteins there too, but synuclein is the main constituent, misfolded aggregated synuclein. And I'm not going to come back to this, but I just want to say very briefly that there are different types of Lewy bodies. So you have the typical brainstem Lewy bodies that have this halo. You have the cortical ones that are a little bit more diffuse. This is a standard hematoxylinase type of stain. Here is the alpha-synuclein stain with a basal forebrain Lewy body and a cortical one. And remember, Lewy pathology isn't just the inclusions in the cell body, it's also these types of neuritic inclusions that are very important and actually more prevalent than the cell bodies in terms of when one looks at the whole brain. So what more do we know about these aggregates? Well, this is a Parkinson meeting, so everybody's very well aware of the fact that Parkinson's disease, before its diagnosis, is preceded by a long prodrome. Then you have a variety of symptoms that develop over time, as shown in this diagram. And the question then is, when do these Lewy bodies occur and where do they occur? And I think everybody knows about the Braak staging idea, or some would say it's a hypothesis. It's impossible to prove in one individual because one can't look at one single individual in a microscope several times. But Heiko Braak and his colleagues has suggested that the pathology over the premotor and the motor phase of disease can be divided into different stages. So initially, at the very beginning in the premotor phase, there's only some pathology in the dorsal motor nucleus of the vagal nerve and in the olfactory bulb, and then gradually it increases. And you can see that these changes, the dorsal motor nucleus of the vagal nerve that innervates the gut, correlate to constipation in the clinical features, and the olfactory bulb Lewy pathology correlates to hyposmia. So gradually these increase, and around stage 3 of the disease, they appear in the substantia nigra, and then as the disease progresses, and you see this in the brain to the right, there's Lewy pathology all over the brain. And that, to some extent, one can argue, coincides in many patients, or some patients, with cognitive decline and other disturbances. So we've now given an introduction. Let's spend the next 13 minutes or so talking about, is Lewy body pathology necessary and sufficient, as the first of our questions? Well, simple answer, actually. Several studies have looked at this. Here's one study from 2012, if I remember rightly. It showed that in patients with LOCK2 mutations or Parkin mutations, only a subset of those patients actually have Lewy pathology. At the very top of the diagram, you can see here are the LOCK2, here's the Parkin, and here at the top, you have actually a few patients with synuclein mutations, and all of them, of course, had Lewy bodies. But the ones with LOCK2 and Parkin mutations, only a subset of them have Lewy bodies. So Lewy pathology isn't necessary for clinical Parkinson's disease. And if we look further at this, you can see that the absence of Lewy bodies, and this is interesting, is associated with reduced risk for certain non-motor features of the disease. And this paper by Kelly et al. Showed that specifically cognitive impairment, anxiety, orthostatic hypotension was less prevalent in those individuals who had motor disturbances but did not have Lewy bodies. So we can conclude, not all clinical Parkinson's disease cases have Lewy bodies. Now, is Lewy body pathology necessary in sporadic Parkinson's disease? And this is a little bit more difficult to address. And I looked at this paper, which belongs or was authored by Michael Henderson, who joined our institute about a year ago as an assistant professor, having worked with Virginia Lee and John Trojanowski. And he mentions in this paper, while the absence of synuclein pathology is rare in PD, we identified 9 atypical PD patients out of 109 total idiopathic PD patients in our database that had a similar Nigel degeneration without, and I stress that, without Lewy body pathology. So I actually emailed Mike yesterday to say, tell me a little bit more about this. Do you have any more updates on this? And he basically says that 8% of the PD patients lacked Lewy bodies, and they were listed as atypical PD, and they didn't progress to dementia. And as he wrote to me, it's possible that they have a rare mutation, or they don't, although they don't have LOCK2 mutations because they were genotyped for the most common LOCK2 mutations. And based on this observation, and I looked in the literature, it's not easy to find these descriptions in modern literature where there's been genotyping, where there's been modern synuclein staining. But based on this, one can say around 90% of sporadic PD cases exhibit Lewy bodies. So how should one take this? Well, some people say that's maybe a lot. And I would actually say, I say high, and maybe you say low. And you say why, and I'll have to say, I don't know. And that's another part of hello, goodbye. So thank you, Beatles, for guiding us through this enigmatic feature of Parkinson's disease when it comes to synuclein and Lewy bodies. So Lewy bodies are definitely not necessary for clinical PD. But are they sufficient? And that's flipping the question the other way around. Well, I think most of you know incidental Lewy bodies occur in 8 to 12%. There have even been higher numbers in some cohorts, up to 20% of people who are neurologically normal and over 60 years old. So not all cases with Lewy bodies exhibit clinical Parkinson's disease. That's very clear. But that's maybe not the end of the story. And there are a few papers one can look at here where people have actually in detail, and this team here headed by Dennis Dixon is one of them, in detail looked at the pathology in these individuals with what we call incidental Lewy body disease because they were neurologically normal. And the substantia nigra neuron density was decreased in these individuals. The TH staining and the striatum seemed to be decreased, not as much as in Parkinson's, but somewhere in between Parkinson's and normal. And finally, also in the epicardium, in the heart, the nerves there, the synuclein, or there was a change in tyrosine hydroxylase immunoreactivity, presumably because of synuclein pathology. And this is not a one-off paper. There have been other papers, this paper here from Roger Curlin and Tom Beach group, showing a very similar observation where there is a gradual, it would seem, a gradual decline of nidal dopamine neurons from normal through incidental Lewy body disease to Parkinson's disease. So, Lewy bodies are neither necessary nor sufficient for clinical Parkinson's disease. That's the answer to that question. But I think we have to throw in this little flag here, that their presence suggests that a pathological process has started. All right, what can alpha-synuclein animal models teach us? Let me start by saying animal models are animal models. They are not the disease. But I still think we can learn important things. And we know from animal models that they replicate Lewy-like pathology with synuclein aggregates and all the other associated proteins. These animals exhibit neuronal dysfunction that can be measured using electrophysiology and through behavioral deficits that are anatomically consistent with the pathology. And eventually they exhibit neurodegeneration and loss of neurons. And there are many types of animal models that have been generated over the years, lots of transgenic mice, over 30 different strains, I think. But I just want to focus on two types of animal models. One is using a viral vector to overexpress protein, either the normal wild-type protein or the mutant protein. And in those cases, you get massive aggregate formation. Here it's done in the nigrostriatal system. And it's also possible to inject so-called PFFs, preformed fibrils of alpha-synuclein, in this case into the stratum, in order to see if one can seed in a prion-like fashion aggregates. And indeed that happens, and one can get aggregates, as you see in the lower right-hand corner here, you can get aggregates in the substantia nigra and also in the amygdala and other regions that are associated with Parkinson pathology. So animal models replicate these features that are so important. But another question is, can you replicate other parts of the Parkinson pathology? So you know, of course, that before you get Parkinson's, there's pathology, as we mentioned, according to Brock's, his idea, there's pathology outside the basal ganglia, and later in disease, widespread pathology throughout the cortex. So the question is, can animal models mimic Parkinson progression, including the non-motor features, starting in the prodromal stages? And here, injections of synuclein fibrils into potential trigger sites, namely the olfactory bulb and the gut, replicate the early Brock stages. So my group has worked on making these sorts of little fibrils, micro-injecting them into the olfactory bulb, looking if we can see what Heiko Brack had seen in the olfactory bulb and anterior olfactory nucleus, and asking the question whether over time that pathology will spread throughout the nervous system from the olfactory bulb of a mouse, wild-type regular mouse. The answer to that question is yes. And here are just some snapshots from 1, 3, 6, and 12 months after the injection. And you see an increasing number of brain regions are engaged with Lewy pathology, synuclein aggregates. And eventually, at the very end of this particular study here, you can see in the brainstem that there are actually aggregates in nigra and locus coeruleus and Ruffin nucleus. Very, very few, but proof of principle that this is sort of replicating what we think happens in Parkinson's disease. And these models show progressive spread of Lewy pathology that follows neural tracts, and they're also associated with olfactory deficits. And that can be detected both with electrophysiology and with behavior. Now, when it comes gut to brain, Heiko Brack has described in the gut there is synuclein pathology, and Ted Dawson and his team, team led by Dr. Koh, have shown that if you make these fibrils that can make Lewy-like aggregates in the brain, you make them in the test tube, you inject them into the duodenum pyloric region of a normal mouse, there is an enhancement of synuclein pathology in the brain. Interestingly, if you cut the vagal nerve, similar to what has been done in humans, as previously as a treatment for severe peptic ulcer, and epidemiologically, cutting the vagal nerve in humans is associated, according to some studies, with decreased Parkinson risk later. If you do that in the mouse, inject the fibrils in the pyloric duodenum region, you get much less pathology in the brain. And importantly, if you do this experiment in a mouse that lacks alpha-synuclein in a genetically modified synuclein knockout mouse, there is no pathology. And it just demonstrates very clearly it's not the fibrils that one is injecting that is, they're not the ones that are spreading. It's actually that they're seeding endogenous synuclein that then spread. So Lewy pathology can spread along the vagal nerve, and animal models have shown us this, and endogenous synuclein is required for this propagation to occur. So what can therapeutic strategies teach us? Well, there are many potential ways to interfere with alpha-synuclein pathology. Here's a review we published a few years ago. One can reduce the amount of synuclein that's made, lowering it at the level of the mRNA. One can try to promote its degradation in the cell, or one can inhibit its aggregation in the cell, or potentially one can promote its degradation or its uptake by other cells once it's outside. And if we think about promoting its degradation outside the cell, one of the most interesting ideas of how to do that is to use antibodies. And I think everybody knows that just a couple of months ago, the first of the phase 2 trials, the one headed by Biogen, showed that their particular antibody, given intravenously every so few weeks, did not meet its primary or secondary endpoints. No change in disease progression. Does the failure of one clinical trial like this definitively invalidate synuclein as a therapeutic target, or does this teach us that synuclein isn't important? I think it's too early to say that. And I know some people think we should give up on synuclein based on this trial, but there are so many other ways that are now being tested trying to counteract aggregation pathology in Parkinson's. Many clinical trials are right now in the pipeline, already underway, using different approaches. And this is a review we wrote a year ago, and there's a list of some trials that one can say in one way or another target synuclein. And I think it is important to wait for these trials. And as I'll describe to you in my very last section, maybe think a little bit more deeper about who the patients that go into these trials should be. Because my last question is, can we identify the synuclein patients, the best ones? Well, it wouldn't be wonderful if one could just look at the clinical profile of patients and pick out those 90% that are likely to have synuclein pathology. A paper that came out in Journal of Parkinson's Disease a couple of days ago says that looking at clinical phenotyping, and they've reviewed a massive literature in a very careful way, essentially says that it doesn't work. You don't get any really meaningful clusters doing it this way. And interestingly, another paper that came out this week that has, I think, 3 of the same authors there, Daniela Berg, Syed Mohammed Fereshtnayed, and Ron Postuma, they focused on clustering or trying to identify clusters from how patients behave in the prodrome. And they are a little bit more optimistic in that review. They're saying that perhaps one can identify those patients that are most likely to have lots of synuclein aggregates as being the ones that are GBA mutant individuals who also have REM sleep behavior disorder. Because I think you all know REM sleep behavior disorder is associated with a 95% risk of synucleinopathy within 15 years. But I still think the ultimate way of doing this is actually to do some kind of biomarker method to identify the synuclein patients. And lots of things have been written about this. This is a great review by a great team. This review is headed by Omar El Agnaf, and they describe in it how one can measure monomers, oligomers, and fibrils of synuclein in a variety of tissues. One can look at skin, salivary glands, gut, and do histology, or can do biochemistry in CSF, in plasma, blood, urine, or in a biopsy. One can use, for example, this RT-QuIC method to amplify any fibrils that are there. And hopefully one day we will have imaging, so we can image the brain or the gut and see who has synuclein aggregates. But this needs the perfect synuclein ligand, and we don't have that. So as my final comment, I want to come back to that initial question. Is alpha-synuclein still the basis of Parkinson's disease? Well, I told you that sometimes I've said no when preparing this presentation. And if you say stop, one shouldn't do trials with alpha-synuclein targeted drugs, I say go, go, go. And that, of course, is another line from the Beatles. Now I can stay, still it is time to go for me because my time is up, and I will actually stay on if there are any questions or comments. But meanwhile, as I go, I want to say to all of you, thank you for listening.